Hi friends, good morning. Next video from Dhruva Edutainment and Vidu classes. That is, what are multiple alleles? Describe multiple alleles with the help of ABO blood groups in man. Friends, what are multiple alleles? Describe multiple alleles with the help of ABO blood groups in man. Friends, this is our question. Okay friends, now see friends, generally a gene has two alternative forms called alleles. Friends, you know it. We have chromosomes in the nucleus. Those chromosomes have alleles at loci. A gene has two alleles. What are those two alleles, friends? Two alternative forms of a gene. Generally, a gene has two alleles. What are alleles, friends? They are the two alternative forms of a gene is called as alleles. But sometimes a gene may have more than two alleles. This is called as what, friends? Multiple alleles. If a gene has more than two alleles, that is called as multiple alleles, friends. That is called as multiple alleles. What is the definition of multiple alleles, friends? See, important definition. A gene with more than two alleles at a same locus on the homologous chromosomes of an organism in a population is called multiple alleles. Friends, multiple alleles are present in the population, friends. This is for MSET and NEET, friends. Multiple alleles are not present in an individual. They are present in the population, friends. They are present in the population. Friends, multiple alleles అనేటివి పాపులేషన్లో ఉంటాయి. ఒక వ్యక్తిలో, ఒక ఇండివిజువల్లో, ఒక ఎనిమల్లో ఉండవు. ఒక పాపులేషన్లో మాత్రమే ఉంటాయి. See friends, what are multiple alleles? A gene with more than two alleles at a same locus. What is locus, friends? It is the position of the gene on the chromosome. At a same locus on the homologous chromosomes of an organism in a population is called multiple alleles. Very, very important definition, friends. Okay, friends. For example, ABO blood groups in man. So, in our syllabus, we have to explain multiple alleles with ABO blood groups. Means, once again, friends, a gene with more than two alleles at a same locus. Once again, friends, a gene with more than two alleles at a same locus on the homologous chromosomes of an organism in a population is called multiple alleles. Is called multiple alleles. Best example what, friends? ABO blood groups in humans. ABO blood groups in human. See friends, multiple alleles cannot exist in a person. Just now I told friends, multiple alleles are present in the population. They cannot exist in a person. Next friends, multiple alleles can exist in a population. These are for MSET and NEET, friends. Multiple alleles can exist in a population. Next friends, the inheritance of multiple alleles is called multiple allelism. So what is multiple allelism? The inheritance, the hereditary of the multiple alleles is called as what, friends? Multiple allelism. Multiple allelism. Okay friends? Now see friends, how can we calculate the genotypes formed from a multiple allele? The number of genotypes from multiple alleles is determined by the following formula. Friends, multiple alleles ఎన్ని ఉన్నాయి, అవి ఎన్ని జీనోటైప్స్ని ఏర్పరుస్తాయి అన్నదాన్ని మనం ఏ ఫార్ములాతో, ఫ్రెండ్స్? దిస్ ఫార్ములా, వాట్ ఇస్ దట్ ఫ్రెండ్స్? n into n plus 1/2 అనే ఫార్ములా ద్వారా మనం క్యాలిక్యులేట్ చేస్తాం ఫ్రెండ్స్. n into n plus 1/2. See friends, number of genotypes from multiple alleles is determined by the following formula. What is that formula, friends? Number of genotypes is equal to n into n plus 1/2. n into n plus 1/2. See friends, where n is the number of alleles. Here you have to remember, friends. Friends, n అనేది ఏంటి, friends? How many alleles are there? Number of alleles. n is equal to number of alleles. Now see friends, here I have given one example. In the example 1, If a gene has two alleles, it forms three genotypes. See friends, if a gene has two alleles, it forms three genotypes. Like example, see friends, its genotype is equal to n into n plus 1 by 2. Here n is equal to what friends? Number of alleles are 2. 2 plus 1, 3 friends. 3 into 2, 6. 6 by 2 is equal to what friends? 3 genotypes. Here you have to see friends, capital T, small t. There are two alleles. They form three genotypes. Capital T, capital T, small t, and small t, small t. Okay friends. Now come to second example friends. If a gene has three alleles, it forms 6 genotypes. See friends, n into n plus 1 by 2. N means what friends? 3 alleles. 3 plus 1, 4. 4 into 3, 12. 12 by 2, 6. So it forms 6 genotypes. Friends, this is for please no need to write in the IP. This is for MSET and NEET. But you have to remember this formula. What is that? N into n plus 1 by 2. N into n plus 1 by 2. Now see friends, ABO blood groups are the best example for multiple alleles. This is for IP friends. ABO blood groups are best example for multiple alleles. The gene of ABO blood types has 3 alleles. The gene of ABO blood group has 3 alleles. Friends, 3 alleles, you know. If a gene has 3 alleles, it forms 6 genotypes according to our equation. These 3 alleles form 6 genotypes. Friends, ABO blood groups are the best example for multiple alleles. The gene of ABO blood types has 3 alleles. These 3 alleles form 6 genotypes. See friends, 1 gene, 3 alleles, 6 genotypes. 1 gene, 3 alleles, 6 genotypes. Here you add one more point friends, 4 phenotypes. 1 gene, 3 alleles, 4 phenotypes, 6 genotypes. This is for MSET and NEET friends. Okay friends. Now come to our ABO blood groups. ABO blood group system was proposed by Carl Landsteiner friends. The system of ABO blood groups were proposed by whom friends? Carl Landsteiner. Carl Landsteiner. Blood groups are also called as blood types friends. Blood groups are also called as blood types. Whenever you hear the word blood type, it indicates what friends? Blood groups. Blood groups are also called as blood types. Human blood groups are of 4 types. Friends, we have 4 types of blood groups. What are they friends? Blood group A, blood group B, blood group AB, and blood group O. Friends, blood group A, blood group B, blood group AB, and blood group O friends. Now see friends, these are also called as phenotypes of blood groups. See friends, A, B, A, B, O are the phenotypes of blood groups. So how many phenotypes we have friends? 4 phenotypes. What are they friends? A, blood group B is a phenotype, blood group A is a phenotype, blood group AB is a phenotype, blood group O is also one phenotype. Now friends, the phenotypes, that is A, B, AB and O types, are determined by the presence or absence of the antigens. Friends, how can we determining the phenotypes? That is blood group A, blood group B, blood group AB and blood group O. For example, you see friends, an individual has blood group A. How you are saying that person has blood group A? That is on the basis of what friends? On the basis of the antigens. Presence or absence of antigens. So on the basis of the antigens, we are determining the blood groups. Friends, these antigens, where do we have antigens friends? Antigens are present on the plasmalemma or cell membrane of the RBC friends, red blood cells. Blood group antigens are present on the plasmalemma or cell membrane of the red blood cells friends. Red blood cells or erythrocytes. These blood group antigens are of 2 types. What are they friends? They are antigen A and antigen B. Friends, how many blood group antigens are there friends? Antigen A and antigen B. What is their location friends? They are present on the plasmalemma of the RBC, red blood cells or erythrocytes. See friends, another important point friends, blood group antigens are carbohydrates. Friends, very, very important. They are not proteins. Blood group antigens are carbohydrates. They are sugar polymers. Blood group antigens are carbohydrates. Friends, sugar polymers, polysaccharides. Very, very important for MSET and NEET. These antigens are also called as isoagglutinogens. What is the another name of the antigens, friends? These are also called as isoagglutinogens, friends. Isoagglutinogens. What are isoagglutinogens? Isoagglutinogens cause blood cell agglutination in the case of incompatibility or unsuited blood transfusion. See, friends, what are isoagglutinogens? Isoagglutinogens cause blood agglutination, blood clumping. When, friends? In the case of unsuitable blood transfusion. That is called as what, friends? Incompatibility. Okay, friends? ఏంటి ఫ్రెండ్స్ మనకి ఐసోఅగ్లిటినోజన్స్ అంటే ఏంటి ఫ్రెండ్స్? ఇవి ఒకవేళ మనం దానికి సంబంధించిన యాంటీజెన్ కాకుండా వేరే యాంటీజెన్ ఇచ్చినప్పుడు ఏమవుతుంది ఫ్రెండ్స్? అక్కడ బ్లడ్ క్లంపింగ్ జరుగుతుంది, బ్లడ్ క్లాటింగ్ జరుగుతుంది, బ్లడ్ కోయాగులేషన్ జరుగుతుంది ఫ్రెండ్స్. సారీ, బ్లడ్ అగ్రిగేషన్ జరుగుతుంది. ఓకే ఫ్రెండ్స్? నౌ సీ ఫ్రెండ్స్, agglutination is the clumping of the cells. It is an antibody antigen reaction. Friends, what is agglutination, friends? Is a clumping of the cells. గుచ్చీకరణ అంటాం ఫ్రెండ్స్, గుత్తులుగా మారుతాయి బ్లడ్ సెల్స్. Agglutination is a clumping of the cells. This is due to what, friends? There is a reaction between whom? Antibody and antigen reaction, friends. Okay, friends? Now, here you have to write reaction, friends. Antigen and antibody reaction. It is a process that occurs if an antigen is mixed with corresponding antibody is called agglutination. See, friends, at the type of blood testing, what we are doing, friends, it is the process that occurs if an antigen is mixed with corresponding antibody. For example, antigen A ఉంది ఫ్రెండ్స్, దానికి మీరు యాంటీబాడీ ఏ కలిపిండి అనుకోండి ఫ్రెండ్స్, it what happened, friends? It called it leads to agglutination. Okay, friends? That is about this is for agglutination or isoagglutination. Please don't write this yellowish one in your IP test, friends. Now see, friends, what are the 4 phenotypes of the blood groups? You know, friends, A, B, AB and O. These are due to presence or absence of 2 antigens. What are they? Antigen A and antigen B. Now, very, very important, friends, these antigens are under the control of a gene located on the chromosome number 9, friends. That is autosomal gene, friends. These antigens, the formation of the antigens are under the control of a gene. So the blood group gene, ABO blood group gene, friends, not RH. The ABO blood group gene is present on the chromosome number 9, friends. So it is autosomal gene. It is autosomal gene. On which chromosome, friends? Chromosome number 9. Now see, friend, blood group A has antigen A on the RBC and antibody B in the plasma. Friends, now you come to our point, friends. For example, a person has blood group A. He has antigen A on his RBC and anti-B, that is antibody B, anti-B antibody in the plasma. Blood group B has antigen B on RBC, anti-A antibody in the plasma. Blood group AB has both antigen A and antigen B on RBC and no antibodies. Sorry, friends. Antigen A and antigen B on RBC, but no antibodies in the plasma. Okay, friends. Now come to O, friends. O blood group people have without antigens. They don't have any antigens on their RBC, but they have what, friends? Anti-A antibody and anti-B antibody in their plasma. See, friends, this whole is present in this chart, friends. See, blood groups, group A, group B, group AB, group O. Come here, antigens, friends. Where do we have antigens? On the RBC. Group A has antigen A, Group B has antigen B. Group AB has antigen A and B. Group O people have no antigens. So they don't have antigens. That is absent. Come to antibodies, friends. They are present in the plasma. Blood group A people have anti-B. Blood group B people have anti-A. Blood group AB people have no antibodies. Whereas blood group O people have antibody A and antibody B. These antibodies are present in the plasma, friends. Antigens are present in the RBC, on the RBC, friends. Okay. Once you see this chart, friends, very, very important. Okay, friends. Now, see, friends, once again. Red blood cell type A, B, ABO. It has what, friends? Anti-B. B has anti-A. AB has no antibodies. O has anti-A and anti-B. Antibody A, anti-B. Now come to antigens, friends. A has antigen A. B has antigen B. AB has both antigen A and B. O blood group people have no antigens. See it, friends. Okay. Now come to blood typing, friends. What is blood typing? What is blood typing, friends? Blood typing is a method to tell us what type of blood we have. What is blood typing, friends? It's a method which tells about us to know about our blood types. Blood typing is a method to tell us what type of blood you have. So it is a method mixing drops of blood with serum containing antibodies. It is a method mixing drops of blood with serum containing antibodies, that is A and anti-B. So if we mix a drop of blood either in the anti-A antibody or anti-B antibody, we can know our blood types, friends. See, friends, here. Please don't write this in the exam, friends. Antibody A, you know it, friends, present in the blood group B and blood group O. Acts against antigen A, present in blood group A and blood group AB. Once again, friends, antibody A, present in blood group B and blood group O, acts against antigen A. This antigen A present in blood group A and blood group AB. Okay, friends. Once again, friends, antibody A, where do we have antibody A, friends? Both blood group B and blood group O, they have antibody A. Acts against what, friends? Antigen A. Where do we have antigen A? In the blood group B and blood group AB. See, friends, antibody B present in the blood group A and blood group O, acts against antigen B and in blood group B and blood group AB. Now see, friends, if a clump or cluster is formed with anti-A antibodies, that blood group is A type. See, friends, now this is very, very important. See, friends, if a clump or cluster is formed with antibody A, that blood is of what, friends? A type. ఏంటి ఫ్రెండ్స్, మనకి ఒక యాంటీబాడీ ఏ తోటి ఒక క్లంప్ లేదా రక్తం గుచ్చీకరణం చెందినట్లయితే ఆ బ్లడ్ ఏ రకమైన బ్లడ్, ఫ్రెండ్స్? ఏ బ్లడ్. బికాస్ ఏ బ్లడ్ హ్యాస్ యాంటీబాడీ బి. హియర్ it is agglutinating antibody A. If a clump or cluster is formed with anti-A antibodies, the blood is of type A. If the clump is formed with anti-B antibodies, the blood is of B type. If the clump is formed with both A and B antibodies, the blood is of AB type. If no clump is produced by either of the antibodies, the blood is of what, friends? O type. Because you know, friends, O type have no antigens. So because of that, there is no clumping. Okay, friends, if no clump is produced with either of the antibodies, either anti-A or anti-B, that blood is of what, friends? O type. See, friends, now A blood group, friends, it is clumping anti-A, but with B no clumping. With anti-B is no clumping with A, nana, because A has anti-A, not anti-B. Okay, like this, see friends. Okay. Now genetic base of the multiple alleles. What is the base for its inheritance? See friends, Bernstein proposed the genetic basis of ABO blood groups. So the scientist who proposed the inheritance of the multiple alleles, inheritance of the ABO blood groups, who is he friends? Bernstein proposed the genetic base of the ABO blood groups. ABO blood groups controlled by the gene I, friends. You know, this gene I is present on the chromosome number 9. This gene I has 3 alleles, IA, IB and IO. Friends, here I stands for isoagglutination. Friends, ABO blood groups controlled by one gene. What is that gene, friends? Gene I. The gene I has 3 alleles. What are they, friends? IA, IB and IO. The genetic base of AB blood grouping is mainly depend on these 3 alleles. That is IA, IB and IO of the gene I. See friends, these alleles are present on the chromosome number 9. You know friends, once again, Bernstein proposed the inheritance of the ABO blood groups. ABO blood groups are controlled by the gene I. Gene I has 3 alleles. They are IA, IB and IO. I stands for isoagglutination. Okay friends, this I gene I is present on the chromosome number 9. Is present on the chromosome number 9. Now friends, the antigens which are present on the RBC are known as isoagglutinogens. Friends, the antigens which are present on the plasma lemma of the RBC are known as what, friends? Isoagglutinogens. The antibodies which are present in the plasma are known as isoagglutinins. One more word to antibodies, friends. They are called as isoagglutinins or isohemoagglutinins. Isoagglutinins or isohemoagglutinins. Very, very important. One more MSET bit, friends. Need bit. These antibodies are IgM antibodies. Very, very important, friends. The antibodies of the ABO blood group are IgM type antibodies, friends. IgM type antibodies. The antibodies of the ABO blood groups are IgM type antibodies. Next, friends, these antibodies of an individual cause agglutination reaction with antigen of another individual. So there is a reaction between antibodies of one individual and antigens of other individual. See friends, receivers antibodies react with the donor's antigens. Friends, receivers antibodies react with the donor's antigens. You have to remember it, friends. Now see friends, the alleles IA and IB are mainly responsible for the production of isoagglutinogens or antigens. So antigens are produced by whom, friends? Who is responsible for the production of the antigens? That is allele IA and allele IB. Out of the 3 alleles, what are those 3 alleles, friends? IA, IB and IO. See friend, IA allele is dominant. IB allele is also dominant. Friends, IA is dominant, IB also dominant. IO is recessive. Friends, IA is dominant, IB is dominant, IO is recessive. See friends, IA and IB alleles are completely dominant over IO allele. See friends, IO is recessive, IA is dominant, IB is dominant. So IA and IB alleles are completely dominant over IO allele. See friends, IO allele is recessive to IA and IB alleles. See friends, IA and IB are codominants. These two are dominant over IO, friends. This is IO, friend, not 10 friends, IO. Example, when IA and IO are present only, IA expresses because IA is recessive. Once again friends, when IA and IO are present in an individual, which allele is expressing its character friends? IA, because IO is recessive. When IB and IO are present, only IB expresses its character because IO is recessive. If IA, IB both are present, that is codominant to each other. So if an individual has IA and IB both alleles, that individual gets AB blood group because they are codominant. See friends, IA is equal to IB. See friends, when IA and IB are present together, they both express their character, that is AB, because IA and IB are codominants. IA and IB are codominants friends. Okay? Now see friends, out of these three alleles, what are they? IA, IB and IO. A child receives one of the allele from each parent. ఫ్రెండ్స్ ఈ మూడు అల్లీల్స్లో ఒక బాబుకి కానీ ఒక పాపకు కానీ సంతతికి మనకి 2 అల్లీల్స్ వస్తాయి. ఒకటి ఫాదర్ నుండి ఒకటి మదర్ నుండి ఫ్రెండ్స్. అవుట్ ఆఫ్ దీస్ 3 అల్లీల్స్ IA, IB and IO, a child receives one of the allele from each parent. Now friends, these 3 alleles are forming 6 genotypes. What are they friends? IA, IB and IO alleles are forming 6 genotypes. See friends, IA, IO. Friends, IA, this is homozygous dominant. IO, this is heterozygous dominant. IB, homozygous dominant. IO, heterozygous dominant. IB, codominants friends. IB, codominant. IO, this is recessive. Homozygous recessive friends. Okay? Now see friends. Now allele from father, allele from mother. See, a child is receiving alleles like this. If he receives IA, its genotype of the child is IA. So his blood group A. Friends, this is homozygous dominant. If a child receives IA from father and IO from mother, its genotype is IO. He also has blood group A. See friends, blood group A people have 2 genotypes, either IA or IO. This is homozygous dominant, this is heterozygous dominant. Now come here friends, if allele from father is IB, mother is IB, so the genotype of the child is IB. So he has what nana? B blood group. If IO, the genotype is IO, so blood group is B. See friends, B blood group people have 2 genotypes. One is IB, another one is IO. Now come to IB. So this is codominants. They have AB blood group. IO, this is purely recessive. They have O blood group. Okay friends. Now see the chart friends once again. IA, A blood group. IO, A blood group. IB, B blood group. I0, B blood group. IB, AB blood group. IO, O blood group. See friends, 1 gene, 3 alleles, 6 genotypes and 4 phenotypes. Okay friends. Now see friends, 1 gene, 3 alleles, IA, IB, IO, 6 genotypes, IA, IO, IB, IO, IB, IO. What are 4 phenotypes friends? AB, O. Now see friends, once again phenotypes and genotypes. Phenotype A, what are the genotypes friends? It has 2 genotypes, IA or IO. B phenotype, B blood group people have B phenotype. What is their genotype? IB, IO. This is homozygous friends, this is heterozygous. Now AB people have IB, only one type of genotype, that is heterozygous. O people have only one type of genotype, that is IO friends. Okay friends. So this is one more chart friends. I have given complete all characters of the blood group friends. See, genotype IA, IO, IB, IO, IO. See, what is the antigen friends? IA, they produce A antigen. IO also produce antigen, A antigen. IBIB B antigen, IBIO B antigen, IAIB A and B antigens, IOIO there are no antigens, absent friends. Now phenotypes, these two produce A blood group, these two produce B blood group, this produce AB blood group, this produce O blood group. See friends, how many? 1, 2, 3, 4, only 4 phenotypes. See friends, antibodies. A blood group people have anti-B, B blood group people have anti-A, AB people have no antibodies, O people have anti-A and anti-B. Now see friends, receive blood from A people receive blood from A and O, friends A and O, B people from B and O, AB people from A, B, AB and O. So friends, AB people are receiving blood from A, B, AB and O. So because of they are called as what friends? Universal recipients. Whereas O people are receiving blood from only O. Now donate blood to A people donate blood to A and AB, B people donate blood to B and AB, AB people only AB friends. They are receiving from all, but they are donating only AB. Whereas O people are universal donors, they can donate blood to A, B, AB and O also friends. Okay. Now see some MSET bits friends. Blood group AB people are more sensitive to infectious disease because these people have no corresponding natural antibodies. Friends, blood group AB people are more sensitive to infectious diseases because these people have no corresponding natural antibodies. They don't have any antibodies friends. Now friends, blood group A, B antibodies are IgM type. These are what friends? These are AB antibodies are IgM type. You know friends, we have 5 types of antibodies: IgG, IgA, IgM, IgE, IgD. Blood group AB antibodies are IgM type friends. Blood group RH antibodies are IgG. Friends, this is for NEET and MSET. AB antibodies are IgM type, RH antibodies are IgG type. Blood groups also provide a good example of codominance. Friends, blood groups are example for multiple alleles. Blood groups are example for codominance. You know friends, AB blood groups are codominants. Which alleles are dominant? A and B are dominant over O. Which allele is recessive? O allele is recessive. Which allele are codominant? A and B are codominant. Universal donor is O negative friends. O negative is universal donor. Universal recipient AB positive is universal recipient. Rarest blood group AB negative friends. Rarest blood group is AB negative. A and B antigens are sugar polymers or carbohydrates. A and B antigens are sugar polymers or carbohydrates. Antigen A has N-acetyl galactose amine sugar. Friends, this is for MSET and NEET. Antigen A has N-acetyl galactose amine sugar. Once again friends, antigen A has N-acetyl galactose amine sugar. Antigen B has D-galactose sugar. D-galactose sugar. See friends, ABO blood group system in human beings is an example of both codominance and multiple alleles. The universal donor and acceptor will be O negative and AB positive. You know friends, universal donor is O negative, universal recipient is AB positive friends. Okay friends, this is about what friends? This is about multiple alleles friends. Multiple alleles. Very, very important friends for IPE, NEET and MSET friends. What is our question friends? Here, what are the multiple alleles? Describe multiple alleles with the help of ABO blood groups in man. Thank you friends. Good day.